Welcome back to this next video in which we are discussing the visual system. Okay, so we've now discussed the central neural pathways that underlie the pupillary light reflex, and we've seen that they involve the medial route of the optic tract. So we've seen that the medial route of the optic tract is going to carry the axons from these photosensitive retinal ganglion cells. And by the way, I had the time to look up how to spell intrinsically, and apparently I was correct. This is how you spell intrinsically. But never mind. So we've seen that that involves the medial route of the optic tract, and that the medial route of the optic tract is going to contain the axons of the photosensitive retinal ganglion cells. We're now going to turn our attention to the lateral route of the optic tract, which goes into the lateral geniculate nucleus, also called the lateral geniculate body. Okay, so in the lateral route of the optic tract, the axons of the retinal ganglion cells that are actually linked up to bipolar cells, which are in turn linked up to rod cells and cone cells, are going to run, and they're going to go into the lateral geniculate nucleus. So, what's actually going to happen within the lateral geniculate nucleus? Well, in the lateral geniculate nucleus, they're going to synapse onto another neuron, which will carry the electrical information onwards. If I draw out one of the lateral geniculate nucleuses here, okay, so what will happen is in will come a axon of a retinal ganglion cell, which, as I say, will be receiving input from a bipolar cell, which is in turn receiving information from rod cells or cone cells, and what will happen is it will synapse onto another neuron here, which will have its cell body in the lateral geniculate nucleus or lateral geniculate body, hence why we call the lateral geniculate nucleus a nucleus. Remember, a nucleus is the name for a collection of cell bodies within the central nervous system. A ganglion is the name for a collection of cell bodies outside the central nervous system, i.e. in the peripheral nervous system, whilst a nucleus is the equivalent thing within the central nervous system. So this is indeed a nucleus. So here's another neuron which will convey the information onwards, and what we want to see is how does this axon now of this neuron that's within the lateral geniculate nucleus actually get to the primary visual cortex. So lateral geniculate nucleus, extremely simple. It is quite simply a relay station. We have already discussed how the pattern of electrical activity in the axons of all of these retinal ganglion cells is going to be able to be used to interpret the visual image. What's happening now is simply a relay station, i.e. one axon goes on to another neuron, and the electrical activity in this first neuron here, the retinal ganglion cell, will be mirrored now in the lateral geniculate nucleus body neuron here. Okay, so the information about the visual image is just now going to be passed on to the primary visual cortex through these neurons that have their cell bodies in the lateral geniculate nuclei. Okay, so the next thing that we need to discuss is how is the information going to get from the lateral geniculate nuclei to the primary visual cortex, and for this we need to actually know where the primary visual cortex is. So that's the next question. Where is the primary visual cortex? Okay, so the primary visual cortex is the cortex that surrounds a fissure known as the calcarine fissure, also called the calcarine sulcus. You can call it either a fissure or a sulcus. So I need to show you where the calcarine sulcus actually is on the brain then. So remember, a sulcus is just an invagination of the cortex inwards, and the calcarine sulcus is actually going to not be easily visible, so it's not one of the sulci that you can see just by looking at the surface of the brain. Instead, it's one that's hidden from view, and you need to take a sagittal section of the brain in order to see it. So what we're going to imagine doing is we're going to take a sagittal section of the brain down the longitudinal fissure, okay, which separates the two cerebral hemispheres. So if I just draw a picture from above to explain what I'm doing. So if we're looking at the brain from above, here are the two cerebral hemispheres, drawn quite badly, but never mind. This is the left cerebral hemisphere, this is the right cerebral hemisphere. We are going to take a sagittal plane right down the middle, split the brain in two, and we're then going to draw the cut open surface, and we'll do it for the right hand side. So we'll be looking in at the cut right hand surface here, and we'll then be able to get a view of the calcarine sulcus. So calcarine sulcus is going to be right at the back of the brain on both sides. You'll have a left calcarine sulcus and a right calcarine sulcus, and it faces into the the longitudinal fissure. Okay, so let me now draw the cut open surface then of the brain. So this sagittal section, and I think actually I'll go over so that we've got more space to do this picture. So this is what you're going to see. So this is the anterior side over here, and then it curves back like this. So let me develop this side. So this is quite a complicated picture, so I want to get it right. So we are seeing the cerebral cortex here that faces into the longitudinal fissure that separates the two cerebral hemispheres. And what I'm drawing now is going to be the corpus callosum, which is the connection between the two cerebral hemispheres, which we will have just cut through. So it's a whole bunch of axons crossing between the two cerebral hemispheres, and we've just cut straight through it here. Okay, so here it goes right like that. So I'll label up little bits here. Whoops. Corpus callosum. Okay, and I'll colour the corpus callosum in turquoise here. So this is a white matter connection between the two cerebral hemispheres, really important. Okay, and this is all cerebral cortex facing into the longitudinal fissure, and back here we're going to see the calcarine sulcus. But before we do that, let me just finish the picture over here. So at the front you'll have the hypothalamus here, and the pituitary gland. So this is the optic chiasm here at the bottom of the hypothalamus. Then you've got the pituitary stalk and the anterior and posterior pituitary, the mammillary body on the right hand side. Of course you have a right and left mammillary body. We're seeing the right one here because we've taken we've taken the sagittal section through, and we're looking at the right hand side. And then that's the end of the hypothalamus here. Behind the hypothalamus we know what you have. You have the midbrain sitting here. Then below that you have the rest of the brainstem. So here's the pons and the medulla, and then the spinal cord. Whoops, below that. And then above the midbrain you're going to have the thalami, and this will be the right thalamus here. Okay, and then I've drawn the thalamus a little bit small. Actually, now enlarge it here. That's probably more the better size, and now I'll correct the picture by colouring in to disguise my hideous mistake here. And let's continue on with the annotations because they're probably quite helpful. So this is the right thalamus that we've got here in purple. Then we've got the midbrain, and I'll colour the midbrain in orange. So we've got the midbrain there in orange. Then below the midbrain we've got the other portions of the brainstem. So we've got the pons here in blue, so that's the pons, and then the medulla below that, and those are the three components of the brainstem here. So the midbrain, the pons, and the medulla, they are the brainstem together. So pons we've coloured in blue, and medulla we'll have in yellow, and of course below the medulla you've just got the spinal cord. So that will have passed through the foramen magnum at the bottom of the skull. Then to add another structure in here, you'll have the fornix coming up and coming down like this. Okay, so here's the fornix going around like that. And let's finish the cerebral cortex at this side. So it will come down like so, and then come up here. And we also need the temporal lobe shown like that. And then finally, let's stick on the cerebellum. So here is the cerebellum, and I'll label that up as well. So we'll colour code the cerebellum in what colour should we have? Red, I think. Okay, so there's a basic picture of what the brain sort of looks like if you take a sagittal section down the longitudinal fissure, and I'll just label up the fornix because that's a fancy structure that you might not be familiar that's involved in memory. It connects the temporal lobe, the medial temporal lobe memory system, the hippocampi, to the diencephalic memory system, which are the mammillary bodies. So really, ideally, the fornix would be coming in above the mammillary bodies, but never mind, it's only a picture. So this is the fornix. Okay, right. So that's just basic neuroanatomy. Let's now see where the calcarein fissure is. So all of this white stuff here, this is cerebral cortex that is facing into the longitudinal fissure. So this is not cerebral cortex that is easily visible. It's cerebral cortex that would be visible to you if you were a little man and you walked into the longitudinal fissure in between the two cerebral hemispheres. Then you would be able to walk on this cerebral cortex. And it's on this cerebral cortex that you're going to find the calcarein sulcus. So the calcarein sulcus is specifically going to be back here. Now, just to put it in context, I'm going to put in another sulcus. So there is another very important sulcus that comes down like this on this medial aspect of the right hemisphere here, and that's called the parieto-occipital fissure. So it connects the parietal and occipital lobes. Parieto-occipital fissure. So I'll just highlight that fissure then we'll have it in yellow. So here is a very important fissure or sulcus, an invagination of the cortex inwards called the parieto-occipital fissure. And remember the important thing about fissures in sulci, that you have cortex lining them. It is truly an invagination of the cortex to expand the surface area of the cortex of the brain, because the cortex is very important for processing. Now, finally, on to where the calcarein sulcus is. It's here. It's a connection that goes up like this, a sulcus that joins onto the parieto-occipital fissure and goes all the way back like so. Okay, so that is where the calcarein sulcus or the calcarein fissure, no matter, depending on what you want to call it, is placed. So here, that's the calcarein sulcus. So this is where the primary visual cortex is going to be. Now, the calcarein sulcus, a few more little facts about it. It's about 1 centimetre deep. So what I mean by that is if I was a little man standing at this entrance to the calcarein sulcus, I could walk inwards for 1 centimetre. So it's 1 centimetre deep. And if we take a little cross section, so let's imagine now going over to a coronal cross section, you'd have something that was like this. Okay, so this is the calcarein sulcus, and here is the cerebral cortex lining the calcarein sulcus. And what I mean by this little statistic is this goes in for 1 centimetre like that, approximately. Okay, so that's a little bit more information about the calcarein sulcus. And the primary visual cortex is going to be this cortex that lines the calcarein sulcus, and also a little bit of this cortex on either side of the calcarein sulcus. So this cortex that I'm now highlighting up in pink there. Okay, all of that is primary auditory cortex, and it approximately goes, primary visual cortex, not primary auditory cortex, and it approximately goes 0.5 centimetres above and below the calcarein sulcus, and it also goes onto the posterior pole of the cerebral hemisphere, i.e. It goes onto the back of the brain, the bit that would actually be visible if you were just looking at the back of the brain, and it goes for about 1 centimetre backwards. Okay, but the important thing that you've got here is that this is the position of the primary visual cortex. It's right at the back of the brain in the occipital lobe, and it's majorly around this calcarein sulcus, which is not easily visible. You have to go into the longitudinal fissure, and it's a sulcus that faces into the longitudinal fissure. Okay, so that's where the primary visual cortex is, and the primary visual cortex has a number of other names. It's also called V1 for visual cortex number 1, and another name for it is the striate cortex, and you will hear it referred to as the striate cortex quite regularly, so I'm glad I've brought this point up. So why is it called the striate cortex? It's because when you actually look at the primary visual cortex histologically, so if you were to take a little section of primary visual cortex and have a look at it under the microscope, what you actually see is a white matter stripe within the primary visual cortex. So normally when you have a look at the cerebral cortex, it looks like this. You've got the grey matter here, which is the cerebral cortex, and then underneath you'll have white matter. Okay, that's what your section of cerebral cortex would normally look like. In the case of primary visual cortex, it looks like this. You have grey matter, and then down the middle of it you have a stripe of white matter. That's unusual for cerebral cortex. It's something that is specifically present within the primary visual cortex, and this stripe of white matter is known as the stria of Gennari. Stria, I believe, means stripe, and Gennari is the person who first discovered it, I would presume. Hence why the primary visual cortex can also be referred to as the striate cortex, the stripy cortex, the cortex with this white matter stripe. Right, so we've now seen where the primary visual cortex is, and I should stress that you have two primary visual cortexes. You have a left primary visual cortex and a right primary visual cortex. We have had a look at the right primary visual cortex here, but you would have the mirror image on the left-hand side. And I'll remind you of what we've discussed earlier. The left primary visual cortex is going to receive all of the information about the right visual hemifield, whereas the right primary visual cortex is going to receive all of the visual information about the left visual hemifield. Okay, and we already know that in each of the lateral geniculate nuclei, that swapping over has already occurred. So the left lateral geniculate nucleus contains all the information about the right visual hemifield, so it makes perfect sense that that's going to link up to the left primary visual cortex, and therefore the left primary visual cortex will get all of the information about the right visual hemifield, and the same for the other side. Okay, so all that remains to see now then is how does the information get from the lateral geniculate nucleus to the primary visual cortex? Well, we can probably just about put this on this picture. So let's say here, or thereabouts is the lateral geniculate nucleus for the right side. I mean, it wouldn't perfectly be in this cross section, but it would be a little bit further back into the piece of paper. But it would be at about that position, just lateral to the midbrain and underneath the right thalamus. So here is the right lateral geniculate nucleus. So this contains all of this information coming about the left visual hemifield, and now these neurons that have their cell bodies in the lateral geniculate nucleus, they need to send their axons to the primary visual cortex. And the way it's going to work is the neurons at the top of the lateral geniculate nucleus, they will send their axons to the portion of the primary visual cortex that is on top of the calcarine fissure, so the top portion up here. And those neurons which have their cell bodies at the bottom of the lateral geniculate nucleus, they will send their axons downwards to the bottom portion of the primary visual cortex that's below the calcarine fissure. So the way it works is kind of like this. Here's one, and here's the bottom connection here. And together these white matter connections, because that's what they are, they're loads and loads of axons, and of course this won't be visible when we've taken this cross section. This is further back inside the white matter of the right cerebral hemisphere here, but this is just a picture to show you how this will work. These white matter connections between the lateral geniculate body and the primary visual cortex, they are called the optic radiation together. So this is the optic radiation. Another name for it is to call it the geniculo-calcarine tract. So it's linking the geniculate body, the lateral geniculate body, and the calcarine fissure, or the calcarine sulcus. So the geniculo-calcarine tract. That's the name, the two names for what we have here. Okay, and I'll colour them in here in orange, and I'll stress again, they wouldn't have been visible in this picture that I've drawn, neither would the lateral geniculate nucleus. It would all be further back into the piece of paper, however, it shows it quite nicely. Okay, so that's the optic radiation, or the geniculocalcarine tract. Now, you can see that there is an upper portion to the optic radiation and a lower portion to the optic radiation. The lower portion to the optic radiation has a name, another name. It's called Meyer's loop. Okay, so the lower portion of the optic radiation is often referred to as Meyer's loop. Right, okay, so the final thing then I want to make sure everyone understands is where visual information from different portions of the visual field is actually going to end up at all the different points along this pathway. So to finish, that's what I'd like to do, and then we'll have a look at what the different lesions to the different portions of the central visual pathway would cause. Okay, so let me draw another picture. So we'll start off with the visual field. So I'm going to divide the visual field up into these different portions. So this is going to be the left visual field and this is going to be the right visual field. So it's going to be as though we are standing here looking at this. Right, so I'm going to colour this in different colours. So this is left, this is right, and I've divided it up into 4 different quadrants. So this is the left upper portion of the visual field. This is right upper portion of the visual field, in turquoise there. This is left lower portion of the visual field in red, and this is the right lower portion of the visual field. So let's follow the central visual pathway backwards. So we'll start off with the 2 eyes. So here's the left eye, and here's the right eye, and we're imagining that we're seeing this from behind. So I'm going to divide the retina again up into 4 quadrants. So this is the superior portion of the retina, this is the inferior portion of the retina. We're imagining we're viewing the eyes from behind. Okay, so now let's think about where information from the different quadrants of the visual field is going to end up on the retinae of the 2 eyes. So let's start off with the left-hand eye here. So let's start with the left lower quadrant. Where's the visual information for that going to end up? Well, hopefully you'll agree that it's going to end up in the the right superior quadrant of the left retina here. And the same is true for the right eye over here. Okay, so all the visual information coming from down here, it'll have to come up at an angle like that, and therefore it'll strike the retina up in this quadrant. Okay? For the opposite quadrant, the right superior quadrant of the visual field, all of the visual information will come down into this quadrant of the 2 retinae, because all the information is coming down like that, and therefore will hit these portions of the retina. Remember, to keep you oriented, we are looking at the retinae from behind. Okay? Then let's do a right lower quadrant here. So all of the information from there will come up at an angle like this, so it will strike the retina in these quadrants, and then finally blue is down there. Okay, so that's where the visual information from these different quadrants will actually end up on the retina. Now let's have a look at where the information from the different quadrants is going to end up as far as the lateral geniculate nuclei are concerned. So the next portion of the visual pathway that I want here is the 2 lateral geniculate bodies. So this is the left lateral geniculate body, this is the right lateral geniculate body. Again, we're going to imagine we're looking from behind, so we're going to divide it up into 2 portions, a superior portion and an inferior portion. The superior portion is going to send its fibres up to the portion of the primary visual cortex that is above the calcarine fissure, and the inferior one will send its fibres into Meyer's loop, and therefore to the portion of the primary visual cortex that is below the calcarine fissure. Okay, right, so let's think about this. Remember, there is crossing over of the visual information so that information from each half of the hemifield will end up on the same side. So information from the temporal hemiretina remains uncrossed, and the information from the nasal hemiretina crosses. And you can see how beautiful this is. You can see that the nasal hemiretina here perfectly correlates to the temporal hemiretina here, and same on the other side with the orange and green ones. Okay, so information from this portion of the retina and this portion of the retina are going to come to the left lateral geniculate nucleus. So I'll say that again. Information from the left temporal hemiretina and the right nasal hemiretina are going to come to the left geniculate nucleus. The fibres from the superior portions of the hemiretinae, the orange fibres here, they're going to come to the superior portion of the lateral geniculate nucleus. So that's a new piece of information here, that the fibres coming from the top of the retina remain at the top all the way along the visual pathway. So they're going to come, the ganglion cells from these portions of the retina highlighted in orange, they're from the superior portions of the retina, they're going to come to the superior portion of the lateral geniculate nuclei. So these are the lateral geniculate nuclei, I just label that up. Whereas the fibres coming from the inferior portions of these hemiretinae, they're going to remain below always, and they're going to come into the inferior portion of the left lateral geniculate nucleus. So that's how the information is going to come into the lateral geniculate nucleus here. The same is true on the right side here. So the right lateral geniculate nucleus will receive information from the right temporal hemiretina and the left nasal hemiretina. And the way it's going to work is that information coming from the superior portions of these hemiretinae will come to the superior portion of the lateral geniculate nucleus as well. So all of that information from the red portions of the retinae about the red portion of the visual field will come up to the superior portion of the right lateral geniculate nucleus. And now the information coming from those blue portions of the hemiretinae, that's the lower portions of those hemiretinae, that's going to come into the inferior portion of the right lateral geniculate nucleus. So this is the way it's going to work. And now, finally, let's stick on the calcarine fissures. So these are supposed to represent the calcarine fissures. This is supposed to be the back of the cerebral hemisphere, the back of the left cerebral hemisphere, now looking into the the longitudinal fissure, and there we're seeing the calcarine fissure, and this is supposed to be the right one here, with the calcarine fissure here. So, the axons then from the superior portions of the lateral geniculate nuclei are going to go to the superior portions of the primary visual cortexes. So, colour this in orange and colour this in red. Whereas the fibres from the inferior portions of the lateral geniculate nuclei, they are going to go to the portions of the primary visual cortex underneath the calcarine fissures, the inferior portion of the primary visual cortex. So colour that in turquoise and colour that in blue. So why have I bothered to do all of this? So that I can show you which portions of the primary visual cortex are going to process information coming from the different portions of the visual field. So on the left hand side, the superior portion of the primary visual cortex will actually process information from the exact opposite quadrant of the visual field, the right inferior portion of the visual field. So it's exact opposite. This is effectively diagonally opposite this. Whereas the left inferior primary visual cortex here, that will be processing information about the right superior visual field. On the right hand side now, the right superior visual cortex, that will be processing information about the left inferior visual field, and the right inferior primary visual cortex, that will be processing information about the left superior visual field. Excellent. So that now is where I'm going to end this discussion of the central visual pathways. We've now seen how the information about visual space is actually going to end up being taken to the primary visual cortex. After that, a huge amount of processing that is not particularly well understood at present is going to occur, and eventually we will end up somehow, by miracle, but ending up with a conscious image of the visual world. It is incredible when you think about it. But I hope what you understand now is that in principle we have delivered enough information to this portion of the brain that in principle it could put that information together to gain an understanding of the visual image. That's what I hope you've understood. Right, so to finish off, I want to talk about different lesions to the visual pathway. The central visual pathway. Specifically, we'll talk about lesions to this central visual pathway delivering information to the primary visual cortices. Okay, so the first simple lesion that I want to talk about is monocular blindness. And this is what occurs if you get a lesion to one of the optic nerves. So I'll try and find a picture. Here we go. If you get a lesion to one of the optic nerves, and we could be talking about loads of different types of lesions. Okay, so it could be a vascular lesion, it could be a stroke that has caused damage to that area, or it could be a cancerous lesion, a tumour might be causing damage to that area. It could be an infectious lesion, you could have got an infection that has damaged that area. It could be an autoimmune attack on that area. There's loads of different ways that you could get lesions to certain areas of the brain. What I mean by lesion is just damage to that area of the brain. Trauma is another good example of how you could get a lesion to an area of the brain. So if you get a lesion to one of the optic nerves, then you're just going to be blind in that eye. No visual information from that eye will be going to the central nervous system. So if you covered up the other eye, then you wouldn't be able to see anything, and that's known as monocular blindness. That's very common sense. The next one that I want to talk about is a lesion to the optic chiasm. What happens if you get a lesion to the optic chiasm? Well, what you get is called bitemporal hemianopia. Now, there's a common, well, not a common, but there's a sort of archetypal lesion that leads to bitemporal hemianopia, which is a cancerous lesion. Specifically, this is very close, this optic tract is very close, the optic chiasm is very close to the pituitary gland. Look at this picture, here's the optic chiasm here, here's the pituitary gland. So if you get cancer of the pituitary gland, a so-called pituitary adenoma, what can actually happen is that can expand forward and it goes right into the optic chiasm, and that's the normal way that people get bitemporal hemianopias because of a pituitary adenoma. So bitemporal hemianopia is quite rare, but one of the classical causes of it is a pituitary adenoma, which is a very rare type of cancer to get. Okay, right. So what actually happens then? Well, you lose the fibres that are crossing over in the optic chiasm. Now let's just study this name for it firstly. Bitemporal, will come back to that. Hemianopia, what does hemianopia mean? Hemianopia, hemi means half. Anopia refers to blindness of the visual field. So hemianopia means you lose half of the visual field. And in bitemporal hemianopia, you're going to lose half of the visual field for both eyes. Now, think which fibres we're going to lose. We're going to lose the fibres that are coming from the nasal portions of the hemiretinae. Okay? Now, what you have to understand is that the information coming from the nasal portions of the hemiretinae is not exactly the same as the information coming from the temporal hemiretinae contralaterally. So for instance, if we take the left temporal hemiretina and the right nasal hemiretina, I've said that they're looking at the same portion of the visual field, which is the right visual hemifield. However, this one is seeing further laterally, further temporally, okay, than this one, because this one's further away. This is closer to the side that it's actually looking at, and therefore it's going to be able to see more that way than this would be able to see, more laterally than this. So the image on this one and this one are not identical. This one's actually seeing an image more laterally, shifted to that side. Okay, and the same is true for the left nasal hemiretina and the right temporal hemiretina. This one is seeing more to that side than this one. So if you lose both the information coming from the two sides of the two nasal hemiretinae, then you're going to lose the information about the most peripheral portions of your vision. So another name for this is tunnel vision. You can see things in the centre, but you can't see as well laterally as you previously could. You lose parts of your lateral vision. Okay, you won't be able to see as much as previously. You'll be able to see things in the centre of your vision, so it'll be like this effectively. You see the tunnel in the centre, but you don't see the things around the outside, because the things over here, okay, in the left visual hemifield, that are very peripheral, they used to be being seen by this left hemiretina here. left nasal hemiretina here, and the things in the right peripheral visual hemifield here, they used to be being seen by the right nasal hemiretina, and they can't be seen by the contralateral temporal one because that's further away. It doesn't see as well. It sees this bit brilliantly, okay? So it will see this bit, but it can't get this bit of information. So you get a more restricted vision. You get this tunnel vision, okay? Right, so you end up then with loss of half of the visual field in each eye. And just to explain the name of this even more, if we think about what the visual field would look like for both eyes in this case, so let's do the right eye and the left eye. So let's have this as the left eye here, and this is the right eye. So this is the left visual hemifield, this is the right visual hemifield, this is the left visual hemifield, and this is the right visual hemifield. For the left eye, you're going to have lost the nasal portion. The nasal portion looks at the left visual hemifield, so you will have lost this portion. So this portion will be blind. This portion you will still be able to see using just the left eye. We're looking at the visual field now, just the left eye. So you'll be able to see the right visual hemifield because you've still got the temporal hemiretina, and that's fine. Now thinking about what the right eye will be able to see information coming from, it's that you've lost the right nasal hemiretina here, which looked at the right visual field, okay, the right visual hemifield. So you've lost this bit. You won't have lost the left visual hemifield because that's viewed by the right temporal hemiretina. So if you look at this diagram, you've got hemianopia in both eyes. You've lost half of the visual field in both eyes, and where is it? It's always the lateral portion of the visual field, the left lateral portion and the right lateral portion. So it's always the lateral portion for each of the two eyes, and therefore we call it a bitemporal. Bi meaning both eyes, and then temporal as another word for lateral. In the case of the head, it's towards the temple. So that's why it's called a bitemporal hemianopia. In both eyes you lose the lateral portion of the visual field, and you end up with this tunnel vision. Okay, right, so that's why that's called bitemporal hemianopia, and that's what occurs when you get a lesion to the optic chiasm. And as I say, the classical thing that causes a lesion to the optic chiasm is a pituitary adenoma. Next one that I want to talk about is homonymous hemianopia. Okay, so hemianopia, we know that that means that you have lost half of the visual field for both eyes. So you're half blind in both eyes. Homonymous means that it's the same half of the visual field this time that is going to be lost for both eyes. In bitemporal hemianopia, it was the opposite parts of the visual field that was lost for each of the eyes. Now you're going to be half blind in each eye, but it's going to be the same side of the visual field that you're blind in each case. Okay, now homonymous hemianopia is going to occur when you get a lesion to the optic tract. So for instance, let's imagine we have a lesion here to the left optic tract. So let's think about what would happen if we had a lesion to the left optic tract. Well, the left optic tract, that's carrying axons coming from the left temporal hemiretina and the right nasal hemiretina, and is carrying information looking at, therefore, the right portions of the visual field. Okay? So, you would therefore get what we would call a right homonymous hemianopia, because you have lost the right portion of the visual field. So drawing this out for both eyes, so let's have the left eye on that side and the right eye on this side. So drawing the visual field for both eyes, so if we covered up each of the eyes in turn and saw which portion we are blind to, let's think about the left eye first. So we've covered up the right eye. What can you now still see? Well, you can only now see things that you were seeing with the nasal hemiretina, because those fibres have crossed in the optic chiasm and they're still in the intact optic tract over here. Okay, so you can see things in the left visual hemifield, but not in the right visual hemifield. So you've lost the right visual hemifield in the left eye. Now let's think about the right eye here. Okay, so which fibres would you have lost? The ones for the right nasal hemiretina, because they crossed over and were in the left optic tract here, and they were looking at the right portion of the visual field. Therefore you've lost that. You won't have lost the left portion because they correspond to the right temporal hemiretina, and those fibres haven't crossed, so they haven't been lesioned. So you can see that you've lost the right visual field in both eyes, hence you will call this a right homonymous hemianopia. You've lost the right visual field, you're blind to the right side of the visual field in both eyes. Okay? And homonymous means it's the same in both eyes. You've lost the same portion of the visual field in both eyes. So be careful, you call homonymous hemianopias after which part of the visual field you have lost, not which optic tract you have lost. A right homonymous hemianopia corresponds to a left optic tract lesion, and indeed a right optic tract lesion would cause a left homonymous hemianopia. So in right homonymous hemianopia you will be able to see the left portion of the visual field but not the right portion of the visual field. In a left homonymous hemianopia you will have lost left portion of the visual field and you will be able to see the right portion of the visual field, and those will occur because of optic tract lesions. Okay, right, the final type of lesion is the one that people find most confusing, which is the concept of a quadrantanopia. Okay, so a quadrantanopia. Now, a quadrantanopia means that you have lost a quadrant of the visual field and you're going to lose it in both eyes. Okay, so there are 4 different possible quadrantanopias corresponding to these 4 different quadrants of the visual field, and they occur because of lesions to the optic radiation. Now remember, the optic radiation is divided into 2 different portions on each side, so there is a left optic radiation and a right optic radiation, but in both cases there's a superior portion and an inferior portion. So when you lesion the optic radiation, usually you just lesion either the superior portion or the inferior portion, and you don't lesion both of them, and this is why you can end up losing just one quadrant of the visual field rather than rather than a hemianopia where you've lost a whole half. So let's think about this. Let's think about what would happen if we lesioned the left, or actually we'll do right superior optic radiation, so this portion. Okay, then we lose all information coming through that, but what information is coming through that? Well, it's the information from the right superior portion of the lateral geniculate nucleus that's going to the right superior portion of the primary visual cortex, and that is carrying information. We have agreed about the left inferior quadrant of the visual field, so you would therefore get a left inferior quadrantanopia. Okay, so you would name it again after which part of the visual field has been lost, not which part of the optic radiation has been lesioned. Okay, to give you another example, let's do the right inferior portion of the optic radiation, the right Meyer's loop. So that connects the inferior portion of the right lateral geniculate nucleus to the right inferior portion of the primary visual cortex. So we're losing this flow of information here. So we'll lose information about the blue portion of the visual field, and that's the left superior portion of the visual field, so that would be a left superior quadrantanopia. Continuing on, if you lesioned the left superior optic radiation, you'd lose the orange portion of the visual field, which is right inferior portion of the visual field, so that would be a right inferior quadrantanopia. If you lesion the left Meyer's loop, then you do lose this information about the turquoise portion of the visual field, and you'd therefore get a right upper quadrantanopia, and that would be in both eyes, so you could cover up each eye in turn and you'd have lost the same portion of the visual field in each. Okay, because remember, by this point we have converged the information coming from both portions, well, from both eyes, from the portions of the retinae that correspond to the same portion of visual space. So we've lost all information coming from that portion of visual space. So that's the quadrantanopias. Okay, so that finishes my coverage of the different lesions to the central visual pathway, those 4 different types: monocular blindness, bitemporal hemianopia, homonymous hemianopia, and the quadrantanopias. I hope you've enjoyed this video on the visual system, and I hope you've learned a lot. Thank you for watching.